Imagine you're trying to understand the total functional dynamics of a massive, sprawling city.
Right, like trying to see how everything connects at once.
Exactly. And you have two incredibly detailed data sets in front of you. First, you've got this beautiful satellite map. You can zoom in and see every single building, every park, every intersection.
Okay, I'm picturing it.
And the second data set is this incredibly dense mathematical database of the city's underground power grid. It tracks the exact voltage and the flow of electricity in real time.
Two very different ways of looking at the same place.
Right. So to truly understand why a specific neighborhood keeps having blackouts, you'd naturally want to overlay that power grid data directly onto your satellite map.
So it's the only way it makes sense.
But here's the catch. The systems holding these two data sets are built on entirely incompatible software architectures. Like physically, you cannot look at them together.
Oh, man. That sounds like a nightmare.
It is. You're basically forced to look at the map on one screen and then stare at this massive spreadsheet of the power grid on another screen and just manually try to guess the connections in your head.
Which is wildly inefficient. And honestly, historically, it's been a massive source of error in biology.
Yes, and that brings us right into the mission for today's deep dive. Because in the context of computational biology, that beautiful satellite map, that's a whole slide image of human tissue, a WSI.
Yeah, and that power grid is the underlying RNA and single-cell genomic data.
Exactly. And bridging that gap is what we are exploring today. We're looking at a brand new open source Python package called LazySlide.
Right. This was just detailed in a March 2026 paper from Nature Methods.
And it is a game changer. Histopathology, diagnosing disease by looking at tissue architecture under a microscope that has been the bedrock of medicine for over a century.
It really has. But it's been tragically siloed from modern molecular biology. I mean, the morphological structures we see with our eyes and the molecular transcripts operating inside those cells, they've been speaking completely different programming languages.
Until LazySlide. We are going to get into some incredibly dense, fascinating territory today. We're talking AI foundation models, cellular segmentation algorithms, and how this multimodal integration is fundamentally changing disease discovery.
It's going to be quite the journey. To really understand why LazySlide is such a necessary breakthrough, we kind of have to start with the sheer physics of a whole slide image.
Yeah, let's unpack this, because a WSI is not like a standard JPEG you have on your phone.
Not at all. We're talking about gigapixel scale. A single slide of a tumor biopsy, when it's scanned at high magnification, contains billions of pixels.
Billions, just in one slide.
Exactly. And they're stored in these highly proprietary vendor-specific formats like .svs or .nindpy. They operate almost like a pyramid of images layered on top of each other at different resolutions.
And if you try to load even a few of those into standard RAM, what happens?
Oh, your standard computing environment will instantly crash, just completely lock up.
Right. And the existing spatial biology toolkits that try to parse these images, I'm thinking of software like QuayPath or CLAM or PathML, they rely on these really fragmented data structures.
Yeah, they do. If you're a genomics researcher working in standard molecular frameworks, you can't just, plug an SVS file into your workflow.
Right. And previous attempts to force this integration were technically pretty flawed. There was this extension called SOPA, right? It tried to bring spatial visual data into the molecular ecosystem.
Yeah, but SOPA operated by forcing users to serialize and physically duplicate these massive WSI files just to append the genomic metadata.
And the disk overhead for that was just catastrophic, wasn't it?
Unbelievable. If a researcher serialized a standard cohort of just a few hundred slides, their disk usage would spike by 5 to 10 times.
Wow, 5 to 10 times.
Yeah. In a high-performance computing environment, that kind of data bloat will literally paralyze a shared storage cluster. You're essentially copying an entire library of encyclopedias every time you just want to add a single footnote.
Which is completely unsustainable. But LazySlide bypasses this entirely with a custom data structure. It's called WSIData.
Right, WSIData. It builds directly on top of this scverse ecosystem.
Specifically inheriting from the spatial data framework, right? Which is basically the current standard for spatial omics.
Exactly. And the way it solves that massive file bloat is brilliant. It acts like a universal library card.
A universal library card. I like that analogy.
Yeah. So WSIData doesn't actually copy the massive file. It just acts as a lightweight pointer. It dynamically accesses the original standard WSI formats right where they currently sit on the server.
So it's not moving the mountain, it's just looking at it.
Exactly. It uses lazy loading, which, by the way, is where the name LazySlide comes from. It pulls only the specific chunk of pixels into memory that the algorithm is actively analyzing in that exact moment.
That saves so much memory. And the scverse integration is huge for user adoption, isn't it?
Oh, it's the critical piece. Because WSIData operates natively in that environment, it utilizes familiar application programming interfaces, or APIs.
So people don't have to learn a whole new language.
Right. It uses the exact same data handling logic as highly popular genomic analysis tools like NData, Scampi, and SquidBee.
So a computational biologist can manipulate these gigapixel tissue images using the exact same Python commands they already use to analyze RNA sequence counts. That is amazing.
It drastically lowers the barrier to entry.
Okay, so let's do a quick recap before I move on. Basically, whole slide images are massive, and existing tools force you to duplicate them, creating terrible data bloat.
Right.
But LazySlide uses WSIData to act as a lightweight pointer, reading the images without copying them, and it plugs right into the tools scientists already use.
Perfect summary.
So a quick question for you listening right now. Have you ever tried to work on a massive video or image file that just completely crashed your computer? Imagine that, but the file holds the key to curing a disease. How would you redesign your system to handle it? It's exactly the problem LazySlide is solving.
It really puts the scale of the problem into perspective.
Totally. So we have this system that can access 1 billion pixels without crashing the server, but simply accessing the image doesn't mean the computer actually knows what it's looking at.
Right. Bunch of pixels is just noise until you give it structure.
Yeah.
That brings us to the actual image processing pipeline.
Yeah, how does LazySlide actually read the slide? Because it has to separate the actual biological tissue from the bright white background of the glass slide, right?
Exactly. And it also has to ignore artifacts, things like dust, air bubbles, or surgical marker ink.
So what's the first step?
It executes this highly optimized computer vision pipeline, heavily leveraging the OpenCV library. It starts by converting the image out of standard RGB color into the HSV color space.
HSV, that's hue,
Yeah.
saturation, and value, right?
You got it.
Yeah.
Because biological tissue is usually stained with hematoxylin and eosin. Those are the rich purple and pink dyes. The tissue itself is highly saturated.
Oh, I see.
So by isolating just the saturation channel, the biological material mathematically pops against the low saturation glass background.
That is so smart. And then from there, the pipeline applies something called Otsu's method, right? Let me see if I can explain this.
Go for it.
So rather than a human just arbitrarily guessing where the cutoff is between tissue and background, Otsu's method looks at a histogram of all the pixel intensities.
Right.
And it essentially finds the mathematical valley between the two peaks. It calculates the exact threshold that minimizes the variance within the tissue class and the background class, giving you a perfect binary mass.
That is exactly right. It's fully automated. Or if the tissue is highly complex, LazySlide can actually bypass Otsu's and deploy a pre-trained deep learning model called GrandQC to generate that segmentation mask.
Okay, so now we've isolated the tissue. What happens next?
Once the tissue is isolated, LazySlide performs tiling. It chops this massive irregular shape into a manageable grid of perfectly uniform image patches, usually 256 by 256 pixels.
Like cutting a giant pizza into tiny, perfect little square slices.
Exactly. And it checks each tile against that mask we just made, dropping any tiles that are mostly background.
Makes sense. Don't waste time analyzing blank glass.
Right. It also uses that GrandQC model to run quality control on a tile-by-tile basis. It throws out tiles where the tissue folded over on itself during preparation or where the scanner just went out of focus. You are left with a pristine grid of high-quality tissue architecture.
And here's where it gets really interesting, because now we take these perfect little tissue tiles and we feed them into vision foundation models.
Yes, the heavy lifters.
The paper tests several of these, right? Models like Uni2, Vircho, H-Optimus, and Gigapath.
Right, these are massive neural networks. They use self-attention mechanisms to analyze the physical morphology of the tile.
So they're looking at things like the density of the nuclei, the thickness of the cell walls, the spatial arrangement of the tissue.
Exactly. And the network translates that visual architecture into a dense, high-dimensional feature vector. Basically, it turns a picture of biology into a complex string of 1,000 numbers.
Turning visual patterns into complex math. But wait, I have to challenge you on something here.
Okay, what's the issue?
If you chop a continuous piece of tissue into a strict grid of tiles, What happens to a cell that is sitting right on the border of two tiles? Doesn't the tiling process literally slice it in half?
It absolutely does. And that introduces a severe spatial artifact.
Because when the algorithm tries to count the cells later, it's going to see two separate, weirdly shaped cells instead of one continuous cell.
Right. And if you are doing downstream single-cell analysis, this double counting entirely corrupts your data matrix. It's been a massive issue in spatial biology.
So how does LazySlide fix it?
It solves it using a highly efficient spatial indexing algorithm known as an STR tree. That stands for sort tile recursive tree.
Okay, break that down for me.
First, LazySlide uses cellular segmentation algorithms like InstantSeg or CellPose to draw exact polygon boundaries around every single nucleus in every tile.
So every cell gets a precise little boundary drawn around it.
Right. Then, the STR tree maps all of those overlapping polygons onto a master coordinate system using bounding boxes. When the tree detects two bounding boxes overlapping across a tile border, it doesn't just jam the polygons together.
Oh, so it's not just a blind copy-paste.
No, it calculates probability-weighted averages for the underlying cell masks and computes the intersection over union. If the mathematical overlap exceeds a certain threshold, the system mathematically merges the two fragments back into a single, cohesive cellular polygon.
That is brilliant. It completely resolves that border cell artifact, and it doesn't require massive computational overhead.
It's incredibly elegant.
Yeah.
And with the cells perfectly mapped and the visual features translated into math, LazySlide then runs something called Leiden community detection.
That's a clustering algorithm, right?
Yes. It looks at the mathematical vectors of all these tiles and groups, the ones that are statistically similar. So without a pathologist ever having to manually annotate a slide.
The algorithm autonomously groups the tiles into biological neighborhoods, like automatically mapping out the mucosa or the lymphoid tissue.
Exactly. It derives the entire organ architecture purely from the mathematical relationships.
Okay, time for another quick recap. So LazySlide segments the tissue using Otsu's method, or AI, then chops it up into pristine tiles.
Right.
Then vision models turn those tiles into mathematical vectors, and that brilliant STR tree algorithm elegantly solves the border cell problem by merging the polygons.
You've got it perfectly.
So here's a question for you listening. If you had to teach an AI to recognize a biological neighborhood, like a specific part of your lung, what visual clues would you tell it to look for first? It's crazy to think the AI is figuring this out just from math.
It is wild, and that autonomy actually pushes us into what I think is the most sci-fi feature of this whole framework, zero-shot learning.
Yes. Let's talk about zero-shot vision language querying. This is where we literally start talking to the tissue in plain English.
It sounds impossible, but because these tissue features have been embedded into mathematical vectors, we can utilize multimodal vision language models.
Models like PLIP, CoNCH, Prism, and Titan.
Right. These models don't need to be trained on a specific task beforehand. They've already been trained on millions of image-text pairs from medical literature. They inherently understand the semantic link between images and text.
So they've basically mapped the visual vectors of the tissue into the exact same mathematical space as the linguistic vectors of the English language.
Exactly.
So how does that actually work mechanically? If I type a word, how does it match it to an image?
It uses something called cosine similarity. Think of it like a compass.
Okay, a compass.
When you type a word, say lymphocyte, the language model translates that text query into a dense vector, an arrow pointing in a highly specific direction in mathematical space.
Got it.
Now, the tissue image has already been translated into its own vector arrow. Cosine similarity simply measures the angle between those two arrows.
Oh, I see. So if the angle is wide, there is no relationship.
Right. But if the angle is incredibly narrow, meaning the arrows are pointing in almost the exact same direction, it means the visual tissue is highly correlated with your text prompt.
That is mind-blowing. And the paper actually demonstrates this with an amazing case study on organ classification.
Oh, this part is fantastic.
Yeah. The researchers took whole slide images from 9 distinct human organs, brain, lung, liver, stomach, all sorts of things. And the foundation model had never been specifically trained to classify this data set.
Zero specific training.
Right. So the researchers just queried the system with a simple text prompt, which organ is this? And using that zero-shot cosine similarity with just a single line of code, the model correctly classified almost all of them.
It's astounding. And you can take that text alignment even further with SAM too.
That's the segment anything model too.
Yeah. Because LazySlide knows the exact coordinates of the tile that scored the highest cosine similarity to your text prompt, you can pass those coordinates into SAM2.
So wait, if I type glomerulus,
The vision language model finds the spatial location of the glomerulus, and SAM2 automatically draws a precise boundary line around it.
completely label-free. That is incredible. If you're a pathologist listening right now, the idea of typing a biological term and watching the software instantly snap a boundary around that exact structure on a gigapixel slide, I mean, that is just staggering.
It accelerates the workflow exponentially.
Okay, recap time. Zero-shot learning allows AI to analyze tissue without specific prior training. It uses cosine similarity to match text prompts to image features.
Right, finding the narrowest angle between the text arrow and the image arrow.
Exactly. And the system can perfectly identify organs or segment-specific cells just by being asked in natural language. So think about this. If you could type any natural language query into a tissue slide of your own body, what exactly would you search for? Just typing a word to find a hidden disease. It's wild.
It really opens up endless possibilities. But if we connect this to the bigger picture, the true paradigm shift happens when LazySlide takes this zero-shot querying and statistically fuses it with single-cell sequencing.
Ah, this is where we finally overlay the satellite map and the power grid.
Exactly. The researchers set up this case study using a dataset from the GTEx-X project. They focused on human arteries.
Right. They had 24 healthy human arteries and 21 arteries suffering from calcification, which is the buildup of calcium plaques in the vessel walls.
And crucially, they also had matched RNA sequencing data for all of these exact same tissue samples.
So they started by deploying the text query mechanism we just talked about, right? They fed the slides into the system and just queried the word calcification.
Yes. The model calculated the cosine similarity between the text and every single tile on the slide. But to generate a single calcification score for the entire patient, the system used a mechanism called top key pooling.
Top key pooling? How does that work?
Well, instead of averaging the scores of all the tiles, which would heavily dilute the signal, since most of the artery actually isn't calcified, top K pooling isolates just the top K number of tiles that have the absolute highest similarity scores.
Oh, so it just takes the most confident hits and pools them together to say, yes, this slide exhibits strong visual evidence of calcification.
Precisely. And those top scoring areas instantly highlighted biological terms like vascular niche and apoptosis, which is programmed cell death, as being heavily enriched in the sick arteries.
But LazySlide goes beyond just finding the visual pattern, doesn't it?
It does. It deploys a module called the RNA linker.
The RNA linker.
Yeah. The RNA linker takes that mathematically derived visual calcification score and bridges it to the RNA count matrix. It runs spatial correlation metrics using Pearson and Spearman correlations to identify exactly which gene expressions are scaling up or down in tandem with the visual severity of the calcification.
So they're fusing the visual data with the molecular data. And to execute this integration across two entirely different types of data, they use something called MoFA, right? Multi-omics factor analysis.
Yeah, running it through the muon framework. MoFA is a statistical model specifically designed to infer latent factors that are shared across multiple data modalities.
So it looks at the variance in the visual feature matrix and the variance in the RNA gene count matrix and extracts the hidden variables driving both.
Exactly. And what MoFA extracted was profound. What did they find?
They found that combining the visual images with the RNA data uniquely identified IL-18 signaling as a key pathway driving the calcification.
Yes. And the crazy part is, if the researchers had only looked at the RNA data by itself, if they only looked at the power grid, they would have completely missed it.
Really?
Yeah. When they took just the RNA data and projected it into UMAP space to visualize the clusters, the healthy patients and the calcified patients did not cleanly separate. The transcriptomic data alone was just too noisy.
But when they used MoFA to fuse the visual morphology with the RNA,
the healthy and sick patient groups separated beautifully on the UMAP projection. The morphology, the physical shape and disruption of the tissue, contained crucial biological context that the sequencing alone simply could not see.
So the physical architecture of the cell and its transcriptomic output are fundamentally linked. By bringing them together, LazySlide uncovers disease mechanisms that are totally invisible if you only look at one modality.
Exactly.
Let's do a quick recap of that because it's so important. LazySlide assigned accurate calcification scores using just a text prompt and top-cap pooling. Then the RNA linker fused those visual scores with RNA sequencing data. And this multimodal approach uncovered the IL-18 signaling pathway's role in arterial disease.
A massive win for multimodal integration.
So listener, think about this. We just saw images and RNA team up to find a disease pathway. What other two completely different types of medical data do you think should be combined to solve medical mysteries?
It really makes you wonder what else we're missing just because our data sets are siloed.
Seriously. But recognizing this theoretical potential is only half the battle. In the highly regulated world of computational pathology, a new framework has to prove it can outperform the heavyweights.
Right. It has to survive benchmarking.
So let's set up this heavyweight bout. The researchers tested LazySlide against the dominant spatial tools. QuayPath, TIA Toolbox, SlideFlow, and others. The test was mouse lung tissue containing breast cancer metastasis,
derived from patient xenograft models. And the objective was to accurately classify diverse microanatomical domains like differentiating the metastatic tumor cells from healthy airways, blood vessels, and stroma.
So what was the score? Who won?
LazySlide completely crushed it. It demonstrated overwhelming superiority in classification accuracy. When equipped with modern vision backbones like 80 Mini, UNI2, or Titan, it easily outperformed QuayPath.
Wow. QuayPath has been the industry standard for years.
Yeah, and QuayPath could only match LazySlide's performance when the researchers intentionally handicapped LazySlide by forcing it to run on an older, non-pathology-specific model like ResNet 50.
That is wild. And what about processing speed?
Transformative. LazySlide's automated tissue segmentation vastly outpaced both the manual and automated workflows within QuayPath. Plus, it accomplished all of this with drastically lower API entropy.
API entropy meaning the code base doesn't require a convoluted, chaotic sequence of commands to execute a task, right?
Exactly. High API entropy is a nightmare for developers. But LazySlide's API is incredibly clean. It requires far fewer lines of code, making it super easy and accessible for small research teams who don't have dedicated software engineers.
But, nothing is perfect. This raises an important question. What is the catch? Where's the structural bottleneck?
Well, the interoperability with the scverse ecosystem is LazySlide's biggest strength, but it dictates the strict use of the ZAR file format.
ZAR. That's a format that stores n-dimensional arrays in compressed, chunked files, right? It allows for that lazy loading we love.
Right. But the cost is realized in high-performance computing environments. Because ZAR chunks the array, it relies on a distributed file structure. A single data set can generate tens of thousands of tiny chunk files and metadata files.
Uh-oh, I see where this is going.
Yeah. If you are running this on a massive supercomputing cluster that uses parallel file systems, like Lustre, you will run into hard inode limits.
Inodes, those track the number of individual files on a server, right?
Exactly. Clusters strictly cap how many files a user can generate to prevent the metadata servers from crashing. So LazySlide's reliance on ZAR means users can quickly hit their file count quota, even if they aren't using that much actual storage gigabyte-wise.
That is a significant infrastructure hurdle.
It is. The developers acknowledge it, and they're actively optimizing the storage backend. They're also aiming future updates at 3D tissue reconstruction, taking these 2D tiles and mapping them into volumetric space, which will push boundaries further, but require even treater data handling.
Okay, final mini recap. LazySlide proved faster and more accurate than industry-standard QuayPath in detecting breast cancer metastases.
Yep, completely dominated.
It requires less code and is highly developer-friendly, but there's a slight limitation with ZAR file count limits on certain computer clusters.
You nailed it.
So for you listening, when transitioning to AI tools in high-stakes fields like cancer diagnosis, how much faster and more accurate does a new tool need to be before we completely retire the old ones?
That's the million-dollar question for hospital IT departments right now.
Truly. So what does this all mean? Summarizing everything we've talked about today, LazySlide isn't just a software update. It's a massive paradigm shift. It breaks the silos. It finally allows the visual tissue architecture to talk directly to the molecular transcriptomic level of our cells.
And that's why everyone listening should care, because this open source tool allows researchers globally to accelerate data-driven, clinically meaningful models. A small university lab can now pull gigapixel images directly from a server without duplication, run them through state-of-the-art vision transformers, and statistically link that visual data to single-cell genomics.
All using clean, concise Python code.
Exactly. It speeds up biomarker discovery and paves the way for highly personalized medicine, and you don't need a Google-sized budget to do it.
It fundamentally levels the playing field.
Yep.
And it leaves me with one final kind of provocative thought for you to mull over. Right now, we're using this zero-shot AI to find things we already know about, right? We type calcification or lymphocyte, and the AI maps it using our current English vocabulary.
Right. Mapping the known world.
But as we continue to fuse this visual map with the molecular power grid, what happens when the AI starts highlighting visual patterns on tissue slides that mathematically correlate with diseases, but which have absolutely no correlation to any named biological structure?
Oh, that is a fascinating thought.
Right. Like patterns that predict disease survival, but we don't even have a word for what we're looking at yet. Are we about to let AI write the next medical dictionary?
I think we are. The transition from applying known labels to discovering entirely unmapped biological phenomena. That's exactly what this multimodal architecture is built for.
The map and the grid are finally integrated. And it turns out, when you look at them together, you discover entirely new pathways hiding in plain sight. Keep questioning the world around you, keep exploring the data, and we will catch you on the next deep dive.